We will now show settings in multiSLIDE that can be simultaneously applied to all the heat maps. We call these global settings. These controls are available in 3 horizontal panels above the heat maps. The leftmost is the scrolling panel. The one at the center is the ordering and filtering panel, and the one on the right is the layout panel. Here we will discuss the scrolling and layout panels. The ordering and filtering panel will be discussed in detail in the next video. First, let's look at the scrolling panel. By default, multiSLIDE displays 40 rows and 30 columns in each heat map when the data is first loaded. For instance, in this data set, there are 73 samples, of which the first 40 are currently displayed. With respect to genes, we had selected 57 of them to visualize. These include the gene ESR1 and the genes belonging to the estrogen signaling pathway. Here, the first 30 of these genes are displayed. In multiSLIDE, there is no restriction on the number of genes and samples that can be visualized in a single snapshot, as we will see. However, when the number of selected genes is several hundreds to thousands, a single omics level heat map alone can take up the entire screen. We therefore provide a way to scroll through the heat maps a few genes or a few samples at a time. This can be done using the up-down or left-right arrows. The scrolling step size, which is the number of genes or samples to move with each scroll, can be specified in the text box. Notice that when we scroll by one sample, the scrolling panel shows that we are now seeing samples 2 to 41. Similarly, if we scroll by 10 genes, this change is also reflected in the scrolling panel. As mentioned before, there is no restrictions on the number of samples and genes visualized in a single snapshot. To change the default values, click the pencil icon next to the values and enter the new value in the text box, and then click the Save icon. We will now change the number of samples from 40 to 73 and the number of genes from 30 to 57. This visualizes all our selected genes across all samples. Now let's look at the layout panel. The layout panel has 4 controls. The first control determines the orientation of the heat maps. In the default view, genes are displayed along columns and samples are along rows. This can be changed to have samples along columns and genes along rows using the radio buttons. The second control is used to change the size of the heat map cells. Here, 5 predefined cell sizes are available. We will select the smallest cell size simply by clicking it. This accommodates many more cells in a single view and can be useful for visualizing larger heat maps. The third control determines the position of the heat maps with respect to each other. By default, all the omics level heat maps are displayed horizontally along a single row. We will now change it by clicking the icon so that the 2 heat maps now appear vertically, one after the other. When there are more than 2 omics levels, they can also be displayed in a grid. Finally, the fourth set of controls can be used to customize the widths of the sample and gene names. Often samples can have very long names. How much of the names are visible can be adjusted using the sample label width. We will now update the gene label width from 100 to 50. Notice that the color bars move to the left with a decrease in the size of the labels.